So this is lesson 8 about osmosis in animal cells. Last lesson was about importance of osmosis in plant cells. So this is all about osmosis in animal cells. So if we take a red blood cells are one of the cells that do not have a nucleus. So they have that typical dumbbell biconcave shape that increases their surface area. And this is really useful for them to carry more oxygen. So a larger surface area means that they have more scope to carry more oxygen. They're red in colour because of the haemoglobin, and the haemoglobin is what helps them to connect with the oxygen to create oxyhemoglobin. And we can see our red blood cell here. If it was in a solution containing less water than itself, what we call a hypertonic solution, just like we looked at with the plants. So in other words, it could be a salty solution or a sugar solution. Then basically what would happen is water would leave the inside of the cell and it would start to shrivel and die. So it's really important that our blood, the plasma that contains our blood, has the correct concentration, an isotonic concentration that's the same as in the inside of our red blood cell. And we can see that the red blood cell here has changed into a kind of star shape. We actually call this crenation, okay, when it changes to a star shape. So homeostasis is a word that we use that's very important. It's about keeping conditions constant in our body. So we need to keep things like our temperature constant at 37.4 degrees Celsius. We need to keep our water levels constant in our body, otherwise we dehydrate. We need to keep salt levels constant. So it's really important to keep the blood constant so that the cells in it do not gain or lose too much water. So this is what homeostasis means. If a red blood cell was in the opposite to a hypertonic solution, if it was in a hypotonic, in other words, it had more water in it than the cell. So if you put a red blood cell into pure water, then again it would die too, because it would absorb water. Water would pass in, it would swell up, and then it would just pop. It would burst. So it's really important that we keep red blood cells in isotonic solutions. So here we can see 3 diagrams. These are on page 39 in your booklet. We know that red blood cells play a really important role in carrying oxygen around the body. And when they're in solution with the correct concentration of water, in other words, an isotonic solution, they have that regular biconcave shape. So here we can see in the middle, this is what our normal red blood cell would look like, the biconcave shape. And we have the same amount of water, H2O, entering the cell by osmosis through our semi-permeable cell membrane of the red blood cell as the amount of water leaving the cell. So this keeps it in equilibrium. If we put our red blood cell into pure water, apologies, let's go back, we can see there's more water molecules outside the cell than there is inside the cell in the cytoplasm. So the water molecules move through the cell membrane, the partially permeable cell membrane, by osmosis from high to low water concentration. The cell starts to swell and we can see the cell lyses here. Lyses is a posh word for pops, bursts, explodes. We use the term it got lysed. The cell has lysed. If we put a red blood cell in a strong, let's say, salt or sugar solution, in other words, what we call a hypertonic solution, then there's more water molecules inside the red blood cell than outside the red blood cell. And so basically water starts to leave our cell by osmosis, going from high water concentration to low, through the cell membrane, and the cell starts to shrivel, and as we said, it has this crenated shape. Crenate means star-shaped. So there is a summary of this on page 40, showing you both in osmosis in plant cells and in animal cells. So if you're struggling to answer the questions on page 37 and 39 to explain about using the idea of osmosis, how these cells gain or lose water or remain the same, have an equilibrium, then please use those 2 pages to help you do so.